Scientists are using CRISPR gene editing to restore vision in people born blind, and the results are giving real hope. People born with Leber Congenital Amaurosis, or LCA, one of most severe forms of inherited blindness, striking children in the first decade of life, now have a treatment option. In 2020, for the first time, a surgeon injected a gene editor directly into a patient's eye in Portland, Oregon. In this video, I'll explain how it works, who it's for, what changed for patients, and the limits you should know. Let me back up and tell you what we're dealing with. At the very back of your eye is the retina, a thin sheet of tissue covered in millions of tiny light sensors called rods and cones. These sensors catch photons, convert them into electrical signals, and beam that information straight to your brain. That's vision. In people with certain forms of LCA, a gene called CEP290 carries a typo, a tiny mutation, and that one typo breaks the entire system. The rods and cones can't function properly. Without intervention, patients go almost completely blind, often before the age of 10. CEP290 mutations are the leading cause of inherited blindness in the first decade of life. And here's the cruel irony. The gene itself is too large to be fixed with traditional gene therapy. The standard delivery method, called AAV gene therapy, is like trying to ship a refrigerator in a carry-on bag. So, for decades, there were no options. Just progressive, irreversible darkness. Until CRISPR. You've probably heard the name CRISPR, but what is it? CRISPR-Cas9 is a GPS-guided pair of molecular scissors. Think about your DNA like a 3 billion letter instruction manual. Every cell in your body has a copy, and somewhere in that manual, there's a typo, one wrong letter causing chaos. CRISPR's job is to find that specific typo and cut it out. Here's how the two pieces work. Guide RNA, the GPS. A strand of genetic material engineered to match the exact location of the mutation. It navigates through billions of letters to find the one spot that needs fixing. Cas9, the scissors. A protein enzyme that follows the guide RNA and makes a precise cut in the DNA at exactly the right location. Once the faulty sequence is removed, the cell's own repair machinery takes over. Now, there's an important distinction to understand here. Traditional gene therapy replaces a broken gene with a new copy from outside. CRISPR edits the original gene. It goes in, removes the bad part, and lets the cell repair it using its own machinery. For the BRILLIANCE trial, scientists developed a CRISPR medicine called EDIT-101. It uses two guide RNAs to flank the mutation in the CEP290 gene, essentially surrounding it, snipping it out, and allowing the gene to function normally again. Now you might be wondering, okay, but how do you get CRISPR scissors into an eyeball? Scientists use a harmless, hollowed-out virus called an adeno-associated virus, or AAV. Think of it as a biological delivery drone. Scientists strip out the virus's own DNA and stuff it full of CRISPR instructions instead. Then, a surgeon performs a delicate procedure called a subretinal injection. They use a tiny needle, thinner than a human hair, to lift the retina so slightly and deposit the AAV directly beneath the photoreceptor cells, right where it needs to be. The virus drifts into the photoreceptor cells, releases the CRISPR toolkit, and the editing begins, right there, inside the living eye, inside the living body. This is what scientists call in vivo gene editing, and the BRILLIANCE trial was the first time it had ever been done in a human being. Patients received a 3-day course of steroids first to calm any immune response. Then the injection, just one shot. The BRILLIANCE trial enrolled 14 participants, 12 adults and 2 children, ranging in age from 10 to 63 years old. All of them had been born with LCA caused by CEP290 mutations. All of them had spent their entire lives with severely impaired vision. Each received a single injection of EDIT-101, the CRISPR medicine, in the eye with worse vision. Researchers then tracked 4 outcomes: best corrected visual acuity, BCVA. Can they identify letters or objects on a chart? Retinal sensitivity, FST. How well does the retina respond to light in darkness? Visual navigation, VNC. Can they complete a mobility course using their vision? Vision-related quality of life. Do they feel their daily lives have improved? Results were published in May 2024 in the New England Journal of Medicine. The findings? Remarkable. 79% of participants showed measurable improvement in at least one outcome. Almost half showed improvements in 2 or more outcomes and reported improved vision-related quality of life. No serious side effects, and it worked. But let me bring this back to the human level. One participant who had never been able to read a single line on an eye chart was now able to find their phone after misplacing it. They could see the small indicator light on their coffee machine and know it was working. Another participant, for the first time in their life, could see the food on their plate. Dr. Eric Pierce, the trial's principal investigator at Harvard's Mass Eye and Ear, described hearing these stories as profoundly moving, noting that these were people who had no treatment options whatsoever. The word promising felt like an understatement. Meanwhile, a separate Penn Medicine trial published in The Lancet in 2024 reported even more dramatic results with a different form of LCA. Some patients experienced vision that was 10,000 times better after treatment. One patient reported being able to navigate outdoors at midnight lit only by a bonfire, something they had never been able to do before. So, who stands to benefit from this technology? Right now, CRISPR eye therapy is specifically being developed for people with Leber Congenital Amaurosis, LCA, caused by CEP290 mutations. Children and young adults with early onset inherited retinal degeneration. Patients with retinitis pigmentosa and, in the future, broader inherited retinal diseases. One researcher at Columbia University, Dr. Stephen Song, put it plainly: CRISPR offers the best hope for restoring retinal function regardless of a patient's specific genetic profile, because it can potentially target any mutation, not just one. That's the paradigm shift here. Traditional gene therapy is one size per mutation. CRISPR is a platform, a toolkit that can, in theory, be reprogrammed for different targets. The BRILLIANCE trial is currently on pause while researchers seek larger collaborators and additional funding to run a proper randomized, blinded Phase 3 trial, the kind required before a therapy can be approved for widespread clinical use. As Dr. Thomas Alleman from Children's Hospital of Philadelphia stated after the trial, the hope is that this paves the road for treatments of younger children with similar conditions and, ultimately, for the many other forms of genetic blindness that currently have no treatment at all. That door is now open. If you found this video valuable, share it. Someone in your life might be waiting for this news without knowing it exists yet. Subscribe for more science that matters. And drop a comment below. I'd love to know. What other medical breakthroughs do you want us to cover next? I'll see you in the next one.